The main function of an mRNA molecule is to what?
Carry translatable message.
Carry a translatable message that can be converted into a protein. Yes. Number 7, transcription is similar to DNA replication because both processes. What?
D? No.
I was going to say B.
Both of them are made in the 5 to 3 prime direction.
Oh, so it was
If you look at, for example, like this right here, on page 10, this.
The leading strand, right?
Well, it's replication is DNA replication. Yes, it would be the leading strand, but even on the lagging strand, it's 5 to 3 prime direction. But on 10.6, that right there, that was in the video. She even mentioned like it's 5 to 3 prime direction. Okay, so it is made in both 5 to 3 prime direction. Number 8, most codons specify a what? Each codon, each 3 letters specifies a what?
Amino acid.
An amino acid. Number 9, energy that drives transcription is provided mainly by
ATP.
ATP. Number 10, anticodons pair with who?
mRNA codons.
Perfect. Number 11, what is the max number of amino acids that can be encoded by a gene that has 45 bases? Anytime we have this kind of question, we need to divide the number by what?
3.
Divided by 3, and that should be 15, but it says what?
The answer's not 15.
So what do we do though?
To the
can
stop
add
codon.
one.
Yeah, no, minus.
Minus 1 because of the
Stop codon
stop
is an
codon. So
amino
minus
acid.
1, so there should be 14.
Mm,
So always divide by 3 minus 1. Okay. Number 12, blank are removed from a new mRNA
Introns.
because they are
Interrupted.
interrupted. Number 13, transcription copies a blank to a complementary blank molecule.
Genome.
Gene or genome?
Genome.
Genome is the entirety of your DNA.
Also, it's just gene.
It's just a gene, which to a complementary what?
RNA.
What kind of RNA?
mRNA.
mRNA, good. And 14, what is the job of tRNA during translation?
It carries amino acids.
It carries amino acids. Remember, that's why the codon and anticodon have to pair up. Okay. And remember, when those amino acids pair up, though, they form a what?
Polypeptide. No.
A
They do form a
phos-
polypeptide, but they form individually to one to the other one is what?
Peptide.
Peptide
Bond.
bond. There we go. Okay, so let's go ahead and move on to the last bit of notes. So this part you might want to record, but
Oh, I was already.
okay, good. So this last part is looking at gene expression. And like we looked at on 10.5, 10.6, those videos that we were watching, gene expression just means how is a protein made, okay? How is a protein made? And we kind of already talked a little bit about this as well when we were looking at page 10.5. If you would, go ahead and read the first bullet point for me, please. Only that first sentence that is underlined. Matthew, please.
Transcription happens for individual genes, not all of your DNA.
Again, transcription only happens for what?
Individual genes.
Individual genes, not the entire thing. All of your DNA is what we call genome, and genes are only expressed in some of them, not all of them. Go ahead and read the rest of the sentence for me, please. Abraham.
Also, not all genes are transcribed all the time or at the same time. Instead, transcription is controlled individually for each gene, or in bacteria, small groups of genes are transcribed together called operons.
So operons are a little bit different just because they're like a bacteria thing, because their DNA is a little bit smaller in terms of how much they have. But again, not all genes are transcribed all the time. It's only really what the cell needs in the body at that specific moment. Okay? Go ahead and read this next bullet point for me, please. Jonathan.
Cells carefully regulate transcriptions, transcribing, I don't know, transcribing just the genes whose products are needed at particular moments.
So, for example, in the body, if you happen to have low blood calcium, your body produces a hormone called parathyroid hormone, but it's a protein hormone. But this hormone is only produced when this happens. So that means that the cells that produce it, they're not going to produce it all the time, only at a particular moment. Okay? You don't have to read the rest, that's the AMP stuff. But it's just a good example. The last thing which says, for example, go ahead and read it for me, please, Abigail.
For example, all cells in your body are able to code for the genes to produce hydrochloric acid. However, not all the cells need to or could without damaging other cells. Therefore, only some of your stomach cells need to activate this gene.
So for example, if we look at this picture below, that line that I've highlighted in pink, that is actually the DNA molecule. But within the DNA molecule, we see, and just this part as an example, we can see that this part right here is essentially a gene, because this is, they're making what? Transcription, or in other words, they're making mRNA. But if you notice, each one of these little things, little squiggles, is mRNA. So you can see that whatever gene this is, whatever gene this is, and whatever gene this is, it's making a lot of mRNA. Does that make sense? In the images and videos that we've seen, we only show one little piece of RNA polymerase making one little piece of mRNA. But what this is showing you that in real cells in our body, there's multiple RNA polymerases that are making multiple mRNAs at one time, because again, we may need it at that particular moment. Like for example, like producing hydrochloric acid or producing parathyroid hormone or whatever the case may be. Okay. Now let's look at mutations. So mutation adds, deletes, or changes nucleotides in a specific sequence. There are two kinds of main classes of mutations. We're gonna look at the second ones in just a second, but they can either be point or frame shift mutations. Point mutations change a single what? Nucleotide. And when they do that, they end up shifting the frame in which the protein or ribosome is reading it. So they can produce sometimes different kinds of amino acids. But what is a substitution? Go ahead and read that one for me, please, Nicholas.
The replacement of one base for another.
What does that mean? Replacement of one base.
They switch out the bases.
They just switch out the bases, whatever that may be. What is an insertion? Go ahead and read that one for me, please, Matthew.
A mutation that inserts or adds nucleotide.
So we're only adding, again, one specific nucleotide. Again, you can add more than one, but if it's just one, then it's called a what? Point mutation. Okay. A deletion, go ahead and read that one for me, please, Abraham.
A mutation that deletes nucleotides.
Again, pretty straightforward. Substitution, substitutes, insertion, inserts, deletion, deletes. Okay. But again, if you change it if it's not 3, if it's 1 or 2, it's going to shift the frame in which the ribosome is reading the mRNA to produce the protein or the amino acid sequence. We'll look at an example in just a little while. Okay. But mutations can be organized roughly into 3 specific categories as silent, missense, or nonsense. So silent mutations are what? Go ahead and read that one for me, please, Jonathan. You don't have to read that part, just silent.
Silent mutation, a mutation that does not change the encode amino acid.
So for example, if we are supposed to code for serine and we get serine, that is a silent mutation. Because again, sometimes we can end up getting the same amino acid even though it's not the exact same letters. And again, we'll see an example of that in just a while. Then missense is a what kind of mutation? Go ahead and read that one for me, please, Abigail.
A mutation that does not change the encoded amino acid.
That does change.
Oh, that does change the encoded amino acid.
So for example, if you were supposed to get serine, but we end up getting glutamine, that is a missense because it changes what we were supposed to get. If it doesn't change it, it's what? Silent. If it does change it, it's missense. But if we actually end up getting a stop codon in the middle of producing a protein, it's called a what? Nonsense, because you're stopping the production of that protein literally in the middle of making it. Okay, it's like giving you a car that's half finished because again, it doesn't make any sense. That's why it's called nonsense. Okay? So let's actually go ahead and practice this by looking at questions 4, 5, and 6 at the bottom of page 10.4. So just like last time, what is the first thing we should do when we have a strip of DNA?
Put it in
Add the
brackets.
Add the brackets or use the lines divided in between what is every 3 nucleotides called on DNA?
Triplet.
Triplet. Codon is for mRNA. So just like that. And if you want, I'm going to give you something else. So when we did that, right? But what do we do after that? After the DNA, we have to convert it into what? mRNA, and after that, we have to make it into
Protein.
amino acids or protein. Good. So this first one, TAC, what should the mRNA be? A
AUG.
U G, and using our mRNA codon charts. What does AUG code for?
Mess.
For MET, because it's the start codon every single time. So what I want you guys to go to do is to go ahead and fill in the rest real quick.
Yo. I think I did this one.
Are you guys done?
Yep.
Alright, so let's go ahead and before we actually look through all of this, what I want you guys to go ahead and do is looking at number 4, the DNA sequence, compare it to number 1 really quickly. Because number 1 and number 4 are the same thing, except number 4 has some type of mutation. We're trying to figure out what kind of mutation it is.
Insertion
Where?
on the T, right there, that
First, second, third, fourth, fifth, or sixth triplet.
6.
So we're only adding this T, so number 1 and 4 are exactly the same except for that. Go letter by letter and look at number 1 and 3, or number 1 and 4, excuse me.
They added a G.
Where did they add a G?
Third codon.
Third triplet?
Oh,
So
triplet.
1, 2, 3, the third triplet, this G is what?
Inserted.
Inserted. So this right here is an insertion. So again, one type of mutation it is an insertion, because it's a point mutation, okay? Does that make sense?
Yes, sir.
Okay. But looking at this, what is this codon going to be right here for on the mRNA? GTC, what should it be, Matthew?
CAG.
CAG. What's the next one, Abraham?
AG.
AGU. What's the next one, Jonathan?
UCA.
What's the next one, Abigail?
AGU.
What's the next one? Or is that it?
Yes. Well, preferably.
One, two. The T, right? So what should this right here be?
A.
Right?
Yep.
Okay, just making sure. So then now let's look at the amino acids. So besides methionine, what should CAG turn into as an amino acid or code for as an amino acid? Nicholas.
Glue.
Glue. And AGU should code for what, Matthew? Sir and or Serene. UCA is what Abraham.
Sir.
And AGU is what Jonathan.
AGU?
Yes.
Sorry.
Serene as well. And we're not going to do anything here because it's only one letter, so it doesn't actually code for amino acid.
I think we missed one.
Yeah, we did.
Which one?
Cause
TAC, the 6th column.
Oh, okay, that's okay. I was wondering.
I
So
thought I
after
was going crazy.
Yeah.
after AGU, oh AGU. Okay, so TAC, what should that one be? Anyone?
AUG.
AUG.
And that's the
Met.
Met as well. There we go.
Yeah.
How does that work?
Yeah.
Does that make sense?
Yeah.
Okay.
also, when you have like just one letter on the end, do you just leave it at
You just leave it because that one letter doesn't code for an actual amino acid. It's just there. Okay. But looking back at number 3, what is the amino acid sequence for number 3? It starts off with met, then,
Glutam, serine, glutam, val,
then.
stop.
So if we look at this, compare this amino acid sequence with this one, again, number 1 and 3, or 1 and 4 are the same pieces of DNA, but 4 has some type of mutation, which was an insertion. So if you notice, we see that we have met, glue, ser. But after this, it is not the same at all. So this is why we also call this a frame shift mutation, because it shifts how that ribosome actually reads it. Because it's not reading the same amino acid, it's reading something completely different. Okay. But a last type of classification of this type of mutation is either a missense, nonsense, or silent mutation. Does it change it?
Yes.
Yes. So which one can it not be?
Silent.
It can be silent. So since it does change it in terms of the amino acid sequence, or does it put a stop sequence somewhere in there? Well, it doesn't really even, it actually takes away the stop. So instead, this would be a, again, missense, nonsense, or silent mutation. Missense, because it's not a silent, because it does change it. Okay. Does that make sense? Okay. Now, the last part of the notes is referring to what caused this mutation to actually occur. We talked about it last time as well. They start with an M and they sound like mutation. These are
Mutagens.
mutagens. Remember, mutagens can be physical, like physical, biological, or chemical. Chemical in terms of things like lead. That's why we don't have lead in our paint anymore. Chemical in terms of things like, physical things like radiation, UV radiation, gamma radiation, X-ray radiation, or biological like viruses. Again, HPV, which is a virus, can cause cancer in some cells. Again, just as an example. Okay. So let's go ahead and turn the page and let's look at question number 15. So 15 we're gonna do together just so that we can really truly understand, again, how to make sure to code for it. Because like I said, question 101, 2, and 3 on the exam is you doing this, okay? So I wanna make sure that you are gonna be really, really good at it, okay? So this question, you do need 2 colors, so please make sure that you have 2 separate colors.
Ladies, calm down.
One at a time. Lady.
I'm giving y'all really pretty colors too. You need some? I'll get you some. I have those pens and they genuinely suck. They don't write. Terrible. And I convinced Mason to buy them too, so he has to struggle with me.
Oh, this one don't break.
Exactly, suck. They actually do.
Are those the,
They're paper mate and they're
but they're the paper
bright.
bright ones?
Mhm.
Yeah, they don't ever work. My sister got me one, or a set for Christmas, and like out of the 5 that I got,
Two?
only 2
Yes.
worked.
Ah.
The white doesn't even work.
Ah.
aren't honestly really crappy.
Yeah.
Okay, so what we're doing again using two different colors. The first one says letter A, deletion of the third nucleotide and the insertion of TC between the 9th and 10th nucleotides. So we're essentially having an original piece of DNA and some type of mutation occurs, which is that over there. So deletion of the third, so 1, 2, 3, and an insertion of the what? TC between the what and the what?
The 9th and 10th.
So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So right here, we're gonna add the T and the C, just like that, okay? And in letter B, it asks us to actually write it out. So that's what we're gonna do, okay? So letter B, this is gonna be my new mutated DNA in this color. So what is my new mutated DNA? What is the sequence? A, G, what? C,
T,
T,
A,
A,
T,
T,
A,
A,
C,
C,
T,
T,
C,
C, G,
A,
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 G, T,
C.
Perfect. And again, you notice I already started putting in spaces in between just so that way, because we're gonna convert it anyways, okay? I'm actually turn it off. Close, turn it off. Bless you.
Sounds so weary.
Then in C, what are we gonna do? What does it say? Go ahead and read it for me, please, Abigail.
What is the mRNA sequence of the old and new strand?
And we need to use
Two
two colors. So again, new is gonna be the magenta, and then the yellow is gonna be the old one. So right here for letter C, I have my old, and then I have my new. I'm gonna do old first, but old is this one right here, okay? So looking at this, what is the mRNA sequence of that old piece of DNA? So again, we're gonna do it together. So what does AGC turn into?
UCG.
UCG. So I'm gonna give you a second just to go ahead and finish it up. Do the new one if you finish it. Again, you're doing the old in one color and the new in another color for mRNA sequence. If you finish it, you can go ahead and move on to the other one, but let's go ahead and do it. Are you guys done or not? Any more time? Okay, I'll give you guys a little bit, another minute, okay? Again, if you're done, then move on to letter D. Okay, so let's go ahead and look at the old DNA. So, the first triplet was UCG. What is the second codon gonna be?
Old.
Of the old, sorry, yes. So it should be UCG and then what?
GAU.
GAU, what's the next codon?
AUG.
AUG, what's the next one?
CUU.
CUU, what's the next one?
CAG.
CAG. Is that it or more? Okay.
Was it right, Abigail?
No, I did something totally different. Hold on.
Abraham, is that what you got? Oh, maybe Jonathan's wrong. I don't know.
Well, no, I don't.
Again, we're looking at the yellow part, the old DNA.
Yeah, that's what I was.
So UCG, what? GAU, then AUG,
Mm-hmm.
then CUU, then CAG.
Y'all are wrong.
I had messed it up.
Yeah,
Oh, y'all thought it was the B?
Yeah,
I-
You're
Yeah.
doing both.
You're doing both. That's the new one.
That's
The
new.
old one was the
old one was the top one.
without
The
the
new one is the
marks.
magenta color.
So you still add the C that you removed.
Okay, that's baby steps. We got it. So new one, we're looking at this strain right here from letter B. So what is this one? It should be
UCG.
UCG.
G.
Then
AUA.
AUA. Then
UGA.
then
GC.
No, GCU.
GCU. Then
UCA.
UCA. And then
G.
G. Okay, that's the old. This is one that was already there. This is the new, which is the one that we brought in from letter B. Okay. Now what are we doing? From letter D, we got to convert it into. Amino acids.
Sequence.
So looking at the old one, what is UCG? Sir. What is GAU?
Ow. I go ow.
The names are there, so you can say them if
Oh.
you can try.
Isoleucine.
Isoleucine, so ILE.
This was old?
Next one, AUG.
I think that was wrong.
No, yeah, that's
Oh
wrong.
wait.
Is aspartic acid.
No.
GAU.
AUA is.
No, you're looking at the old. We're looking at the new one.
Stupid letters.
What is GAU?
GAU is aspirin acid. Aspartic acid.
ASP, okay. And then AUG is? That's why we use the colors.
The colors just make it worse.
Really?
Yes.
It's just you formatted weird. You started with the new and didn't start with the. You started with the old and didn't end
Yeah,
with the
why
new.
would you start with the new? Old is first, new is second, and it's just like the.
That's what I did. Oldest first.
I got confused and then just.
Uh-uh.
I did it right. It was just in a different
Because
format.
it
So I just
goes
changed
new,
colors.
old, new, old, new, and it should go old, new, old, new.
That's where I first saw it, but.
It's fine. No,
Okay,
it's
okay. So what AUG is met, what is CUU?
LU.
LEU or leucine. CAG,
Glue.
glue or glutamine. And the new mutated DNA, UCG is what? Serine still, right?
Yeah.
What is AUA?
Oh, that's isoleucine.
Isoleucine, good.
Iso-
And then UGA is? UGA.
Stop.
Stop. So what do we do? Stop, because if we were ribosome, we would stop. So looking at this, the first, again, we're only looking at a section of DNA. We're not looking at the entirety because we don't have a start codon, right? So if you look at it, the first one is what? Serine, which is good, right? It's the same one. Then what happens afterward?
They change.
They change completely. Not only that, but the next one from a start codon literally ends with a
Stop.
So looking at question, number E, explain what this could mean for the organism. What does that mean? First off, this is a what kind of mutation? Nonsense, because it produces a stop codon. But what does this mean for the organism though?
They don't need it anymore.
That what?
They don't need it anymore.
Not that they don't need it,
They don't know.
they don't produce it. So this protein will not be produced. Why? Because it what?
Stop.
It stops. This protein will not be produced which could mean what?
It could form a mutation.
Well, it's already a mutation, but what could this mean for the organism? This protein that we're producing, for example, could be hemoglobin. Hemoglobin is a protein that holds onto oxygen. So that means that it could, not true, but it could cause the organism to die. Does that make sense?
Yeah.
Because
If-
mutations could, again, doesn't say yes, it says it could cause it to die.
Even if it's one mutation? Like
Even if it's one, yes.
That's insane.
Because again, you see, look, what does it do?
It stops.
It just stops. So we're not even making the protein that we're supposed to be making,
Fine.
which could cause
Thank you.
the organism
I like the water bottle.
Thank you. I got from the dollar store.
to die.
What caused this mutation?
So what cause these mutations? Things called
Mutagens.
mutagens. And that's it. It's just one word, even though it's all one word, it's just one.
How do you spell it? I want to make sure I got it right. Mutagens?
Mutagens. Okay. Are we good with this? Can I move on from it?
Mm-hmm.
Okay, I want to go back really quickly just to emphasize this, because mutations can be what? Deleterious, which means that it could cause the organism to die. Another word for it is like lethal. Okay? Or it could be what?
Neutral.
Which means it has no phenotypic effect, which means that you don't really see a result from that mutation. This could be more something like silent mutations. Okay, yes.
What does phenotypic mean?
Phenotypic is the physical characteristics of an organism. So for example, like hair color, eye color, but also the production of a specific protein, okay, if it's present or not. But mutations in what kinds of genes?
Homeotic.
Homeoetic genes, if mutations occur in those, it can cause the body plans to essentially be shifted or mutated or look completely different, okay? And it says they can also have deleterious, which could cause an organism to die, okay? Not delirious, but deleterious. And mutations are the starting point of, especially if they are not neutral, not deleterious, but beneficial, okay? And I'll give you a couple of examples of mutations in homeoetic genes. So we have examples of humans with tails. So these are examples of humans, again, with tails.
What about the horns?
Even then, sometimes those are more like keratin, like skin cells that as kind of like nails, but within specific like parts of your body. Yes.
Why with tail?
Again, if you look at it, humans, again, as an embryo, we technically what?
We have a tail.
We have a tail, but the genes, homeoetic genes, turn off and on to say, hey, actually, we're humans, so we don't need tails. Let's go ahead and cut that stuff away. But when there are mutations in those genes, it says, do we need it? I don't know, do we? And then they are confused about it, so then you end up being born with one. Like you see those other images, okay?
We were monkeys.
We evolved from
Chimpanzees.
Fish.
monkey-like organisms.
Fish?
Monkey-like
I've
organisms.
seen people fish. We crawl out as little fish out
That's,
of water.
that's not real.
Oh,
But another example of this is webbing between fingers and webbing between your toes.
I have webbed fingers.
You do?
No,
Okay.
I could swim.
I can't hear you because Abigail's like
Extra toes.
extra toes. That would be polydactyl. But regardless, that is still an example of a mutation in those homeoetic genes.
Polydactyl.
Oh my gosh.
Poly means more than one,
I have
but
little
it's meaning more than 5.
Yeah, that's the connection.
Oh.
So again, we do have like the skin between our fingers, but this webbing can sometimes confuse bones, or it can just fuse the tissue between it. You can have like, there's different kinds of, like for example, it's called syndactyly. So syn means together, so that means your fingers are like fused together. Or there's, again, as you can see, like
At the
in
bottom.
like this is syndactyly where their fingers are fused together. That's fake, but you can see like they just have extra skin in between their fingers.
Is it like treatable?
Cut it.
Usually they can cut it, but sometimes people don't have it cut. But like when it comes to like children like this, you can see most of the time they do end up getting surgery, which again, it depends on if the bones are fused or not. But if they're not fused together, then they can just cut it, but then they do have to go in and do a skin graft.
Yeah, I figured.
But again, examples of mutations in what kinds of genes?
Homeo.
Homeoetic genes, because homeoetic genes code for specific body plans, which in our case, like no tails, right?
I saw one of this girl, her legs were fused together, and she was called like a mermaid.
It's very similar to like syndactyly, yes.
I thought it was the coolest thing when I was little.
Mermaid is a crazy name.
So what I want you guys to go ahead and do is, we have plenty of time left, but we're gonna go ahead and do this part first, and then we're gonna do the Kahoot. Who's not gonna be here tomorrow?
We might
Oh
be
wait,
back.
no, yeah, I'm going JA.
I thought we were gonna be back
JA?
by that time.
We should be back by the time, that's the thing. Cause they said we should be back.
But I'm not gonna have it here, because we're gonna set up for the Fall Fest.
Ah.
So yeah, we're gonna do the Kahoot afterwards. So what I want you guys to go to do is do the first part, okay?
So like, part 5 or part 1?
This right here.
Oh, okay.
You guys are literally doing it what we did before, but on your own.
I like it.